Today for you listening, we are taking apart the human immunodeficiency virus, HIV.
The apex predator of the viral world. I really think that's the only accurate way to frame it. It defined the late 20th century, and it absolutely continues to define global public health in the 21st.
So here is the mission for this deep dive. We aren't just doing the standard high school health class overview. We are going straight into the engine room today.
We have to. To understand it, you have to see how it works.
Exactly. We're going to break down the molecular mechanics of how this thing actually hijacks a human body. We've got the virology, the genetics, the evolutionary history, and the pharmacology all lined up.
And I think we need to start with the name. Not just HIV, but the broader family name. Lentivirus.
Right, the source text makes a really big point of this distinction. It's a specific genus of the Retroviridae family, and the root word there is lenti.
Which is Latin for slow.
Which honestly feels a little counterintuitive, right? I mean, for a pandemic that just exploded so aggressively in the 80s, why call it slow?
Because it plays the long game. There are retroviruses that infect you and cause tumors almost immediately. The text refers to those as transforming retroviruses.
Okay.
They are oncogenic, aggressive, and fast. But a lentivirus is different. It's cytopathic. It infects you and then, well, it waits.
It sets up camp.
Yeah, it establishes a chronic persistent infection that lasts a lifetime. The text is very unambiguous about this. Once infected, there is no natural clearance.
You just never get over it.
Never. The human immune system never wins on its own. It's basically a slow-motion car crash that takes years or sometimes decades to finally total the car.
And that total collapse, that's what we call AIDS, right? Acquired immune deficiency syndrome.
Exactly. It's the terminal stage where the immune system is so heavily dismantled that it can't even fight off a common cold, let alone fend off cancer.
And the scope of this is just massive. The text mentions that AIDS is one of the most significant public health problems of the 21st century.
Undoubtedly.
But it also frames the development of highly active antiretroviral therapy as one of the major achievements in modern medicine. So it really is this epic story of a perfect killer and humanity's scramble to outsmart it.
That is exactly the dynamic.
Okay, let's get visual for a second. If you and I were to shrink down and look at this virus under an electron microscope, what are we actually looking at? Because the source has these images, figure 44.1. It's clearly not just a blob.
No, it has a very specific and elegant architecture. First off, it's spherical. It's about 80 to 100 nanometers in diameter.
Pretty standard size for a virus.
Yeah, but the giveaway, the thing that tells a virologist immediately this is HIV and not some other retrovirus, is the core.
The core?
Inside the sphere, there's an electron-dense cylindrical core. The text explicitly calls it a bar-shaped nucleoid.
A bar-shaped nucleoid.
Right. Most other retroviruses have a concentric round core, but HIV has this distinctive cylindrical bar shape inside.
Like a bullet inside a chamber.
That is a very fitting analogy,
Yeah.
And inside that bullet, inside that core, is the payload, the genetic material. Now, HIV is an RNA virus. Specifically, it's single-stranded RNA. But here is a fascinating structural detail. It's diploid.
Wait, diploid? That usually refers to human cells having two sets of chromosomes. You're saying the virus has backup copies?
Yes. It carries two identical copies of its RNA genome in every single virion. It basically travels with a spare.
That feels like massive over-engineering for a virus. Why would it need a backup drive?
It's an insurance policy. If one strand gets damaged by cellular defenses, or if the virus wants to do some complex genetic recombination during replication, which it does, it has a spare to work with.
Yeah.
It just maximizes the chances of survival.
Okay, so we have this bullet-shaped core with a redundant backup drive. Let's look at the code itself. The text lists the genes, and you've got the standard package that all retroviruses have. The big three genes, GAG, POL, and NV.
Let's knock those out first, because they're the foundation. GAG encodes the core proteins. This is the structural stuff.
Scaffolding.
Exactly. The capsid protein CA, the matrix MA, and the nucleocapsid NC. GAG basically builds the physical house.
Okay, got it. What about POL?
POL stands for polymerase. This gene encodes the enzymes, the heavy lifters of the virus. This single gene gives us reverse transcriptase, protease, and integrase.
These are the actual tools the virus uses to replicate and integrate its code.
Right. And then NVO. NV encodes the envelope. These are the glycoproteins on the surface of the virus, some specific GP120 and GP41.
The spikes.
Think of them as the grappling hooks the virus uses to latch onto a human cell.
Okay, so GAG-POL and NVILD, the physical virus, that seems pretty standard for retroviruses. But the source material specifically says HIV is much more complex than those fast-acting, transforming retroviruses. It has these accessory genes.
This is where the engineering gets terrifying. HIV has 6 additional genes. And these aren't just structural spare parts.
Right.
These are highly specialized regulatory genes and offensive weapons used to actively fight the host's immune system.
Let's run through them because the names seriously sound like a roster of comic book villains. You've got TAT, REV, NEF, VIF, VPR, and VPU. Let's start with TAT.
CAT stands for transactivation. Basically, this gene is a turbocharger. It wildly amplifies the expression of all the other viral genes.
So it speeds things up.
Dramatically. Without TAT, the virus replicates, but it's very inefficient. With TAT, it goes into absolute overdrive. The TAT protein binds to the newly forming viral RNA and just floors the gas pedal on transcription.
Okay, so TAT is the gas pedal. What about REV?
REV is the logistics manager. It stands for Regulator of Expression of Virion Proteins.
Catchy.
Virologists love their acronyms. Here's the problem REV solves. When the virus makes its RNA deep inside the host cell's nucleus, that RNA needs to get out into the cytoplasm to be turned into actual proteins.
Otherwise, they're just blueprints locked in a filing cabinet.
Precisely. REV is the courier. It facilitates the export of unspliced viral RNA out of the nucleus. Without REV, the blueprints never leave the office and the virus never gets built.
Gas pedal and logistics.
Yep.
Got it. Now let's talk about NEF. The text singles this one out. It says it's necessary for pathogenicity. It even mentions that SIV, the monkey version of the virus without NEF, doesn't actually cause disease in monkeys.
MEF is arguably the most insidious gene in the entire payload. It acts as a stealth cloak.
How does it hide?
It actively downregulates CD4, but crucially, it also downregulates MHC class I molecules on the surface of the infected cell.
Whoa, hold on. Let's break that down for the listener. MHC class I is like the ID badge a healthy cell wears to show the immune system what's going on inside, right?
Exactly. It's the window through which the immune system's killer T cells look to see if there's a virus inside.
So if a cell is infected, it puts a piece of the virus on that MHC class I badge to signal for help.
Yes, to say, hey, I'm compromised, destroy me. By removing MHC class I from the surface, NEF effectively pulls the blind shut.
That's chilling.
The T cells patrol by looking for infected cells, and the HIV-infected cell just looks completely blank. It hides the active infection from the body's immune surveillance.
So it literally blinds the security cameras. That is incredibly sinister.
It is, and it works perfectly. And then you have VIF.
VIF, the bodyguard.
Yes, the defender. See, human cells aren't completely helpless. We have a natural cellular defense mechanism, a protein called ApoBec3G.
ApoBec3G.
All right, this is a cellular genome editor. When a retrovirus enters the cell, ApoBec3G tries to attack it by causing massive mutations in the viral DNA.
It tries to scramble the virus's code.
It basically tries to mutate the virus to death. It's brilliant defense. But HIV has VIF, viral infectivity factor.
And what does it do?
VIF binds directly to ApoBec3G and basically tags it for the cellular trash can, the proteasome, before it can do any damage to the virus.
It assassinates the internal security guard.
Before the guard even draws its weapon. It's an elegant countermeasure.
It's just amazing to me that this tiny strip of RNA has a highly specific tool for every single defense we throw at it. What about VPR?
The PR is the transporter. It helps get the newly synthesized viral DNA through the nuclear pore and into the nucleus. But it has a secondary function that's fascinating. It arrests the host cell cycle in the G2 phase.
It freezes the cell.
It pauses the cell's normal division cycle in a state that happens to be the most metabolically favorable for viral replication. It essentially holds the cell hostage at peak production capacity.
Wow. And finally, VQ. The text mentions this has a very specific function regarding the release of the virus.
VPU is the degrader. It promotes the degradation of CD4 inside the cell.
Wait, why would it want to destroy CD4? I thought CD4 is the receptor the virus uses to get into the cell in the first place.
It is, and that's exactly the problem. If an infected cell is busy making thousands of new virus particles, and that cell still has CD4 receptors on its surface, the new baby viruses, which have the GP120 keys on their surface, will just bind to the CD4 on the cell that just made them.
Oh, I see. They get trapped on the surface.
Exactly. They get stuck to the Velcro. VPU solves this by destroying the CD4 molecules inside the cell before they ever reach the cell surface.
It removes the Velcro so the new viruses can float away.
It cleans up its own exit route to ensure the new generation can escape and infect other cells.
Okay, so to quickly recap this payload for the listener, you have a gas pedal with TAT, a logistics manager with REV, a stealth cloak with NEF, a bodyguard assassinating our defenses with VIF, a transporter that freezes cellular time with VPR, and a demolition crew clearing the exit with VPU.
And another detail the text notes about this genome reading frame overlap. To fit all these complex tools into such a tiny physical space, the virus uses the exact same stretch of RNA to code for different proteins just by shifting how it's read.
That is the ultimate sign of biological efficiency. Every single nucleotide is weaponized.
And that is why it is such a formidable enemy. It's prepared for almost everything the host cell can throw at it.
Which leads us perfectly into the concept of evolution and mutation. Because even with all this brilliant machinery, the virus isn't static. The text talks about something called the quasi-species concept.
This is a critical concept for understanding why HIV is so hard to beat. Remember the enzyme reverse transcriptase?
The one encoded by the POL gene, the tool it uses to copy its RNA into DNA.
Exactly. Well, it turns out reverse transcriptase is a very sloppy worker. It makes mistakes.
Typos in the genetic code.
Constantly. Human DNA polymerases have a proofreading mechanism to fix errors. HIV's reverse transcriptase does not. And because the virus replicates so incredibly fast, making billions of copies, these typos accumulate rapidly.
So what's the result of that?
The result is that if you take a single infected person today, they don't just have one uniform strain of the virus in their body. They have a massive swarm of closely related but genetically distinct viral genomes.
And that swarm is the quasi-species.
Yes.
So to be clear, my HIV might look different from your HIV, but also my HIV today looks completely different from my HIV next month.
Exactly. It is constantly shifting. And this is why it matters so much clinically. Drug resistance. If you hit that swarm of billions of viruses with just one drug, chances are that somewhere in that swarm, one specific virus has a random typo that makes it resistant to that drug.
And since all the susceptible ones die off,
that one resistant mutant survives, it replicates, and suddenly the entire swarm is completely resistant to your drug.
And I assume this same sloppy replication is what makes developing a vaccine such an absolute nightmare.
It is the primary reason we don't have an HIV vaccine. The target is always moving. The envelope proteins, those GP120 spikes that our antibodies look for, are the most variable parts of the entire virus. By the time your body makes antibodies for one version, the virus has already changed its code.
It's like trying to hit a bullet with another bullet while both are changing shape.
Precisely.
Let's zoom out for a second from the single patient to the actual family tree of this virus, because you mentioned earlier that it's a lentivirus, and the text makes it clear that these exist in a lot of different animals, right?
They do. Lentiviruses are actually quite common in mammals. They're in cows, horses, cats, sheep. The prototype lentivirus, the first one really studied, was the Visna virus in sheep.
And what does that do?
It causes a slow, progressive pneumonia and essentially brain rot in sheep. But the closest relatives to human HIV are the SIVs.
Simian immunodeficiency viruses. Simian meaning monkeys and apes.
Yes. SIVs are found all over Africa in African green monkeys, sooty mangabeys, mandrills, chimpanzees. But here is the really interesting ecological dynamic. In their natural hosts, like the African green monkey, SIV usually does not cause disease.
Really? So the monkey carries the virus but doesn't get simian AIDS?
Correct. The monkey and the virus have evolved a biological truce over millions of years. The virus replicates, but the monkey's immune system doesn't overreact and destroy itself. They just live together.
But I'm guessing that truce doesn't hold if the virus jumps species.
It absolutely does not. If that SIV jumps to a new species, say from an African green monkey to a rhesus macaque, which is an Asian monkey that hasn't evolved with the virus, the rhesus monkey's immune system collapses. It gets simian AIDS and it dies.
Because the new host hasn't negotiated that evolutionary treaty yet.
Exactly. And tragically, that is exactly what happened with humans. HIV is the direct result of a cross-species transmission, the zoonotic spillover.
Now, the source clarifies that there are actually two distinct types of HIV, right? HIV-1 and HIV-2. Did they come from the same monkey?
No, they came from completely different primate species in different events. HIV-1, which is the global killer, the one responsible for the worldwide pandemic, came from chimpanzees. Specifically, a strain called SIVD-PZ.
Of HIV-2.
HIV-2, which is less pathogenic, it progresses slower, and it's mostly localized in West Africa, came from sooty mangabeys. SIV-ism.
So the virus jumped into humans at least twice.
Multiple times, actually. But those two lineages established themselves in the human population.
And when did this jump actually happen? Because I think in the public consciousness, most people associate the start of HIV strictly with the 1980s.
Right. The epidemic was officially recognized in 1981 by the CDC. But the jump from primates to humans happened decades earlier. Molecular clock analysis tracing back those mutations we talked about suggests HIV-1 entered humans in the early 20th century.
How early are we talking?
Estimates generally hover around 1930, but some genetic data pushes it as far back as 1908.
1908. So it was circulating in humans for over 70 years before we even noticed it.
Yes. It likely simmered in highly localized rural populations in Central Africa. This aligns perfectly with the cut hunter theory.
Where a hunter gets infected through a cut while butchering a chimpanzee for bushmeat.
Exactly, blood-to-blood contact. But for decades, it stayed local. The transmission chains would just burn out. It wasn't until the social and economic changes in the mid-20th century that it exploded.
What kind of changes?
Rapid urbanization. The building of new highways, facilitating travel changes in sexual behavior, the introduction of unsterilized injection campaigns for other diseases. All of these modern factors provided the perfect highway for the virus to expand out of rural areas and become a global epidemic.
It's a perfect storm. And within HIV-1, we have these different groups and clades. It's like reading a taxonomy of misery in the textbook.
It really is. HIV-1 is broadly divided into groups M, N, and O. Group M stands for Main, and it earns that name because Group M is responsible for over 90% of all global infections.
And then Group M is split up even further.
Yes, into subtypes or clades labeled A through K.
The text mentions clade B and clade C specifically as being important.
They are the most epidemiologically significant. Clade B is the dominant strain in North America and Europe. So historically, because of where the funding was, most of the early research and drug development was done on clade B.
Okay, and clade C?
Clade C is the dominant strain in sub-Saharan Africa and parts of India, which means clade C actually represents the vast majority of the global burden of the disease.
Okay, so we know what this machine is. We know its origins and how it mutates. Let's talk about the actual invasion. How does it physically get into our cells? We mentioned the GP120 grappling hooks earlier.
Right. The primary receptor, the lock that those hooks fit into, is called CD4.
And CD4 is found on what kind of cells?
Primarily on helper T cells and macrophages. These are the central commanders and the heavy infantry of the human immune system.
So it doesn't just attack random tissue, it specifically targets the very cells whose only job is to coordinate the defense against viruses.
Exactly. It decapitates the army's leadership. That's why the collapse is so total.
Yeah.
But here is a crucial detail. Binding to CD4 isn't enough to get inside.
You need something else.
Yes. When GP120 binds to CD4, it causes a structural shape change in the viral envelope. This change exposes a second binding site. The virus needs a co-receptor. It needs a second key to turn the lock.
And the source names two specific co-receptors, CCR5 and CXCR4.
Correct. And this biological distinction dictates the entire course of the disease. Strains of HIV that use the CCR5 co-receptor are called macrophage tropic.
Because they infect macrophages easily.
Right. And these CCR5-using strains are almost always the ones that establish the initial infection. They're the transmission strains.
And the other one, CXCR4.
Later in the disease process, as the quasi-species evolves inside the body, the virus often mutates to use CXCR4 instead. These are called T-tropic strains. When this switch happens, the decline of T cells usually accelerates drastically.
Now, why is CCR5 so famous in the virology world? Because the outline specifically points out genetic immunity related to this receptor.
This is one of the most incredible discoveries in the history of HIV research.
Yeah.
Virologists noticed that certain people who were repeatedly exposed to the virus simply never got infected.
Their cells were just rejecting it.
Yes. It turns out that some individuals, mostly of Northern European descent, have a specific genetic mutation in their CCR5 gene. It's called the Delta 32 mutation.
Delta 32. And the text says this is a homozygous deletion, meaning they inherited this broken gene from both their mother and their father.
Exactly. Because they have this homozygous deletion, their cells simply do not manufacture the CCR5 receptor. It is missing from their cell surface.
And since the transmission strains of HIV absolutely need CCR5 to open the door,
the virus just bounces off. These people are highly resistant, virtually immune to HIV infection.
They are literally locked doors to the virus.
Precisely. And understanding this naturally occurring immunity leads us directly to the most famous medical case in HIV history, the Berlin patient.
Right. The cure case. We have to talk about this.
Timothy Ray Brown. He was a patient living in Berlin who had well-controlled HIV, but he tragically developed acute myeloid leukemia, a blood cancer.
So he needs a bone marrow transplant to survive the leukemia.
Right. His doctors, led by Gerald Hutter, had an incredibly brilliant, if risky, idea. They needed to find a bone marrow donor that matched Timothy's tissue type. But they specifically searched the registry for a donor who also had that homozygous CCR5 delta 32 mutation.
Wow. So the plan was to basically wipe out his cancerous HIV-infected immune system and replace it entirely with a new immune system that is genetically immune to HIV.
That was the gamble. They used radiation and chemo to destroy his native bone marrow and then transplanted the donor stem cells.
And it worked.
It did. After he recovered from the transplant, he stopped taking all of his antiretroviral HIV medication. And they waited. And the virus never came back. It was undetectable in his blood, in his tissues, everywhere.
He was cured.
He was the first person ever cured of HIV. It provided the ultimate proof of concept that a cure is biologically possible.
But the text is quick to note that this isn't an easily scalable solution.
Well, definitely not. You can't just give all 35 million people living with HIV a bone marrow transplant. The mortality risk of the transplant procedure itself is extremely high.
So it's not a public health intervention.
No, but it points the way forward. It tells us that if we can artificially replicate that effect, say with CRISPR gene therapy, to delete CCR5 in a patient's own cells, we might actually be able to cure them without needing a donor.
Okay, let's step back to the typical infection process. I want to talk about the Trojan horse aspect mentioned in the outline. We always focus so heavily on the T cells, but the text highlights monocytes and macrophages as major players.
Yes, macrophages are literally the big eaters of the immune system. They patrol the tissues and engulf foreign pathogens. HIV easily infects them via that CCR5 receptor. But here's the trick. Unlike the T cells, which are killed quite rapidly by the virus, HID doesn't always kill the macrophage.
So the macrophage survives the infection.
It survives and it becomes a mobile factory, a reservoir.
A hiding spot that walks around.
Exactly. And macrophages are designed to travel. They can cross the blood-brain barrier. They migrate deep into the lungs and the lymph nodes.
So the body's own defense cells are carrying the virus behind friendly lines.
They are the ultimate Trojan horse. They disseminate the infection into protected tissues like the central nervous system, which directly leads to the neurological complications we see later in the disease.
Okay, let's walk the listener through the actual timeline of the war inside the body. Someone gets exposed, they get infected. What happens? The text breaks this down into 3 distinct stages, and figure 44-4 illustrates it beautifully.
Stage 1 is primary infection, often called the acute phase. This usually happens about 3 to 6 weeks after the initial exposure.
What's happening on a cellular level?
The virus is replicating explosively. There is a massive spike in the amount of virus in the blood. We're talking millions of copies per milliliter. This is peak viremia.
And during the spike, how does the patient feel?
They usually feel awful. They develop a syndrome that looks a lot like infectious mononucleosis, fever rash, swollen lymph nodes, fatigue, sometimes a sore throat.
And their CD4 count.
It drops sharply. The virus is slaughtering the T cells in the gut and the blood.
But then the acute symptoms usually go away, right? People feel better.
Right, because the immune system finally mounts a massive counterattack. The CD8 killer T cells wake up and start destroying the infected cells. The viral levels plummet down to a much lower stable plateau.
And the text calls this plateau the viral set point.
Yes. The set point is crucial. It dictates how fast the disease will progress. A high set point means you progress to AIDS quickly. A low set point means you might have years. And during this time, the CD4 count does rebound a bit, though usually never back to its pre-infection baseline.
And this transition leads us right into stage 2, clinical latency. The outline calls this the long grind.
It is a grind. This is the highly deceptive phase. The patient might feel completely fine. They're often entirely asymptomatic. And this phase can last a decade or more if they aren't on treatment.
But inside, it's not actually latent, is it?
Not at all. Clinical latency is a misnomer. The virus is replicating furiously. The text provides a staggering statistic here. It estimates that up to 10 billion virus particles are produced and destroyed every single day.
10 billion a day.
Every day. It's a massive unseen churn. The body is pumping out new CD4 T cells from the bone marrow and the thymus as fast as it possibly can to replace the casualties. And the virus is killing them just as fast.
A literal war of attrition.
Exactly. And because of this hyperactivation, the half-life of a CD4 cell in the bloodstream drops from a normal lifespan to about 1.6 days.
That is exhausting just to think about. Your body fighting at maximum capacity for 10 years straight.
And eventually the body just runs out of resources. The regenerative capacity of the immune system eventually burns out. The bone marrow can't keep up. The architecture of the lymph nodes gets destroyed.
And the virus begins to win.
Yes, the CD4 count starts its relentless slide down year after year.
Which inevitably leads to stage 3, the collapse. AIDS?
AIDS is strictly defined by clinical metrics. It's diagnosed when the CD4 count drops below 200 cells per microliter of blood. Normal is around 1,000. Or it's diagnosed when the patient develops a specific AIDS-defining opportunistic illness.
At this point, the fortress walls are completely down.
Yes, the immune system has effectively failed. And without treatment, death usually follows within 1 to 2 years of entering this stage.
And it's usually not the HIV itself that kills the patient, right? It's the opportunistic infections.
Correct. The virus just removes the guardrails. The opportunistic infections are what take advantage.
The outline lists these, and they aren't some rare, exotic superbugs. These are things we encounter all the time.
Exactly. They are opportunists. In a healthy body, they are handled effortlessly. You wouldn't even know you were exposed. But in an AIDS patient, they are lethal.
Let's talk about some of the major killers the text highlights, starting with protozoa.
Toxoplasmosis is a major one. It's a common parasite. You often find it in undercooked meat or cat litter. In you or me, it does absolutely nothing. But in a patient with AIDS, it reactivates and causes severe brain abscesses and lesions.
Wow, and the gut.
Cryptosporidium. It causes chronic, relentless, watery diarrhea that can lead to severe dehydration and wasting.
What about fungal infections?
The most common early sign is candida.
Thrush?
Right, thick white patches in the mouth and down the esophagus. But the real fungal killer, historically, is Pneumocystis jirovecii.
That causes pneumonia, right?
Yes, PCP pneumonia. Before modern prophylaxis, this was one of the leading causes of death. The lungs basically fill with fluid and the patient suffocates. There's also Cryptococcus, which causes a terrible fungal meningitis in the brain.
And bacteria. Tuberculosis seems to be the absolute heavyweight here globally.
Mycobacterium tuberculosis is the leading killer of HIV patients worldwide, period. The risk of developing active TB disease increases 20-fold if you have HIV.
A 20-fold increase.
Because the immune macrophages that normally wall off TB in the lungs are completely dysfunctional. And here is a terrifying clinical wrinkle. Because HIV destroys the immune response, a standard TB skin test might actually show up as negative in an AIDS patient.
Wait, really? Because they don't have enough immune cells to mount a reaction to the test?
Exactly. They have raging TB, but the test is negative because they have no immune system left to cause a little bump on the arm. It makes diagnosis very difficult.
That is brutal. What about other viruses?
Herpes simplex infections become chronic and severe. But CMV cytomegalovirus is particularly nasty in late-stage AIDS. It can infect the retina.
Causing blindness.
Yes, CMV retinitis. It literally destroys the visual field. It can also cause severe colitis in the gut.
And then there are the malignancies, the cancers. Because the immune system is also responsible for hunting down mutant cells, right?
Right. Immune surveillance for cancer stops. The most iconic cancer associated with AIDS is Kaposi sarcoma.
If you look at the medical photos from the early 1980s from New York and San Francisco, you see patients with these dark purple spots all over their skin.
That is Kaposi sarcoma. It's actually a vascular tumor, a cancer of the blood vessels. We now know it's caused by a secondary virus, human herpes virus 8 or HHV8.
Was this common before HIV?
Incredibly rare. It was mostly seen in elderly Mediterranean men. But suddenly in the 80s, it was 20,000 times more common in this young population. 20,000 times.
Than other cancers.
Non-Hodgkin lymphoma is very common. An aggressive cervical cancer driven by HPV is much more prevalent in women with HIV.
Now we have to touch on the neurologic disease. Because we mentioned the Trojan horse macrophages carrying the virus into the brain. The text says 40 to 90% of patients have neurological symptoms.
It is a profound component of the disease. The virus enters the brain very early on. It doesn't infect the neurons directly, but it infects the microglia, the brain's resident macrophages.
And that causes inflammation.
Massive inflammation and the release of toxic viral proteins, which leads to neuronal death. Clinically, this manifests as AIDS dementia complex, or ADC.
And this isn't just minor forgetfulness.
No, it is devastating.
Okay.
It starts with memory loss and an inability to concentrate, but it progresses to severe motor deficits, tremors, and eventually can lead to a completely vegetative state.
The outline also mentions something called vacuolar myelopathy.
Yes, that affects the spinal cord. Tiny microscopic holes literally form in the spinal tracts, leading to leg weakness, spasticity, and loss of bowel control.
And the pediatric cases.
In children born with HIV, the neurological impact is tragic. Severe developmental delays, encephalopathy, failure to thrive. It stunts the developing brain.
It really attacks every single aspect of the human experience, doesn't it? Memory, sight, movement, breath.
It comprehensively dismantles the person.
So how do we actually catch it in time? Let's move to diagnosis. How do we test for this machine?
The gold standard for decades was serology. which means we look for the antibodies the patient's immune system makes against the virus.
Not the virus itself.
Right. We use an EIA, an enzyme immunoassay, as the initial screening test. It is highly sensitive. It rarely misses an infection.
But if it's positive, you have to confirm it, right?
Always.
Yeah.
You must confirm a positive EIA with a Western blot.
Okay, break down the Western blot for us. The source lists specific bands like P24, GP41, and GP12160.
The Western blot physically separates the actual viral proteins on a gel strip. When you apply the patient's serum, you are looking to see if their antibodies react to those specific protein bands.
So the P24 is the core protein from the GAG gene.
Right. And GP41 and GP120 are the envelope proteins from the NV gene. To definitively diagnose HIV, the lab needs to see reactions to specific combinations of those bands. It proves the antibodies are specifically targeting HIV components.
But there is a major catch with antibody testing, the window period.
This is a critical public health concept. It takes time for the human body to manufacture detectable levels of antibodies, usually anywhere from 4 to 12 weeks after the initial infection.
So if I have a high-risk exposure on a Friday and I go get an antibody test on Monday,
the test will come back completely negative, even though you are actively infected and highly contagious. The antibodies just aren't there yet.
so how do we close that window? The text mentions newer generation tests.
Yes, fourth-generation tests are the standard now. They look for the antibody, but they simultaneously look for the viral antigen, specifically the P24 core protein itself.
Ah, so it looks for a piece of the actual virus.
Exactly, because the P24 antigen spikes in the blood during that acute phase weeks before the antibodies show up. This combination testing dramatically shortens the window period to just a couple of weeks.
Okay, so a patient is diagnosed. From there, we track two vital numbers to monitor the war inside, the viral load and the CD4 count.
My favorite analogy for this, and it's a classic in infectious disease, is a train heading towards a cliff.
Okay, lay it out.
The CD4 count is your distance to the cliff. It tells you how much damage has already been done. If your CD4 count is high, say 800, you are miles away from the edge. If it's below 200, you are right on the precipice of AIDS.
And the viral load?
The viral load, which measures the exact number of RNA copies in your blood, tells you the speed of the train.
How fast you're moving toward the cliff.
Right. If the viral load is high, millions of copies, you are rushing towards that cliff at full speed. The disease will progress rapidly.
But if it's suppressed?
If the viral load is undetectable, thanks to medication, the train is effectively stopped on the tracks. You won't go over the edge.
That is a brilliant analogy. It makes total sense. Let's talk about the global scope of this train wreck, the epidemiology, because the numbers in this textbook are staggering.
They are humbling. As of the publication of this source, there were over 35 million people living with HIV globally.
And where is the epicenter? Where is it hitting the hardest?
Sub-Saharan Africa. It accounts for roughly 68% of all global cases. In some regions, the prevalence in the adult population is just heartbreakingly high. Whole generations have been affected.
Figure 44-7 has a map that breaks this down. It shows Sub-Saharan Africa is bearing the brunt, but it also notes significant numbers everywhere else.
Yeah, North America has about 1.5 million cases. The Caribbean has around 240,000. It is a true global pandemic.
And how is it primarily spreading? I mean, we all know about sexual transmission.
Sexual transmission is the primary route worldwide. Anal intercourse carries a particularly high risk because the mucosal lining is thin and rich in target immune cells. But the textbook highlights a massive amplifying factor, other STDs.
Sexually transmitted diseases like syphilis or herpes.
Yes. If someone has an active STD that causes genital ulcers or inflammation, it recruits thousands of CD4 cells to the area to fight that local infection.
Which provides more targets for the HIV virus.
Exactly. The presence of another STD increases the risk of transmitting or acquiring HIV by up to 100-fold.
100-fold? That's an insane multiplier.
It drastically changes the transmission dynamics in populations with high STD rates. Then, of course, there is blood transmission.
Blood transfusions used to be a major fear.
Used to be, yes. But today in developed nations, the blood supply is aggressively screened with nucleic acid tests. The risk from a transfusion is infinitesimally small now.
But injection drug use?
IDU remains a major vector. Sharing needles injects the virus directly into the bloodstream, bypassing all mucosal defenses.
What about needle sticks in a hospital setting? I know medical workers worry about this constantly.
It is a huge source of anxiety, but statistically, the risk is actually quite low. After a percutaneous exposure, a needle stick from an infected patient The risk of acquiring HIV is about 0.3%,
Okay, that's much lower than hepatitis C, for example.
much lower.
Uh.
But it's still treated as a medical emergency requiring prophylactic medication.
And the last major route is mother-to-child transmission, vertical transmission.
This is one of the greatest tragedies, but also one of our greatest scientific successes. Without any intervention, the risk of an HIV-positive mother passing the virus to her baby, either in utero, during the trauma of birth, or via breastfeeding, is anywhere from 13 to 40%.
But with treatment.
If we can get the mother on antiretroviral therapy and suppress her viral load, that risk drops to less than 2%.
That's phenomenal.
It is. We have the tools to virtually eliminate pediatric AIDS globally if we can just fix the logistics of getting the drugs to the mothers who need them.
Let's talk about the virus outside the body for a second, disinfection. Because in the early days, there was so much hysteria about casual contact, catching it from a toilet seat or a handshake.
And that fear was completely biologically unfounded. HIV is an enveloped virus. It is actually incredibly fragile outside the human body.
Because of that lipid envelope.
Right. If that oily envelope dries out on a surface, the virus dies instantly. It cannot infect you. It is easily inactivated by standard disinfectants. 10% bleach, 50% ethanol, 35% isopropanol, or just extreme changes in pH.
The text notes, it is not killed by 2.5%. Tween 20, though.
Right. Tween 20 is a mild detergent. It's not enough to disrupt the envelope. But the point is, you cannot get HIV from a toilet seat, a doorknob, or a handshake. It requires direct fluid-to-fluid transfer.
However, the source gives a specific warning for laboratory workers, the rule of 3 for experimental inactivation.
Yes. While it's fragile on a dry table, it is highly formidable in a test tube or syringe. The rule of 3 reminds scientists why it requires strict biocontainment. 1, the virus persists indefinitely in hosts. 2, it has high mutation rates. And 3, it is transmitted by body fluids.
So you treat every fluid sample as if it's lethal.
Always. Universal precautions.
Okay, let's finally get to the hope. Segment 8, fighting back, the treatment.
The development of antiviral drugs for HIV is honestly one of the greatest collaborative scientific achievements in human history. We have built a massive arsenal of drugs that target entirely different steps of the viral life cycle.
Let's break down the classes of these weapons, starting with the oldest NRTIs, nucleoside reverse transcriptase inhibitors.
Drugs like AZT or tenofovir. Remember, reverse transcriptase is building a DNA chain out of nucleotide blocks.
Yeah.
NRTIs are basically fake building blocks.
Decoys.
Right. The enzyme grabs one of these fake blocks and tries to add it to the chain. But the fake block is missing the chemical hook needed to attach the next block.
So the chain terminates, it jams the zipper.
Exactly. Viral replication stops right there.
Okay. Then we have the NNRTIs, non-nucleoside.
These also stop reverse transcriptase, but via a completely different mechanism. They don't pretend to be blocks. They bind directly to the enzyme itself and physically distort its shape.
Like throwing a wrench straight into the gears.
Yes. They just gum up the machinery. Efavirenz is a classic example of this class.
Next class, protease inhibitors, or PIs.
Remember that the virus initially translates its genes into long, continuous protein chains that are useless. They need to be precisely chopped up into individual working parts.
And the viral protease enzyme acts like a pair of scissors to do that cutting.
Exactly. PIs like ritonavir block those scissors. The result is that the cell still produces new virus particles and releases them. But because the proteins were never cut properly, the new virions are immature and completely non-infectious.
You're duds.
Totally harmless duds.
And then we have integrase inhibitors.
These block the integrase enzyme.
Yeah.
So the virus can make its DNA, but it can never splice it into the host's chromosomes. It stops the invasion of the library.
And finally, fusion and entry inhibitors.
These work on the outside of the cell. They literally block GP120 from binding to CCR5, or they prevent the viral envelope from fusing with the cell membrane. They just bar the door.
So we have all these distinct weapons, but as the text emphasizes, the strategy is the most important part. HART, highly active antiretroviral therapy.
The golden rule of HIV medicine, you never, ever use just one drug alone.
Because of the quasi-species, the swarm.
Exactly. If you give a patient just AZT, the virus will mutate a workaround in a matter of weeks. The resistant strain will take over.
So what's the HART strategy?
You hit the virus with 3 different drugs from at least 2 different classes, all at the exact same time. A cocktail.
So maybe 2 fake blocks and a wrench in the gears.
Right, because the statistical probability of a single virus particle randomly developing 3 specific mutations to beat 3 different mechanisms at the exact same moment is practically zero.
It traps the virus mathematically.
It does. It suppresses the viral replication so thoroughly that the viral load drops to undetectable levels. The train stops. The immune system has breathing room to recover, and CD4 counts rise.
People with undetectable viral loads can live totally near normal lifespans.
And crucially, if you are undetectable, you cannot transmit the virus to your sexual partners. U equals U. Undetectable equals untransmittable.
That is amazing. But we have to be clear, it is not a cure.
No, it is a highly effective management strategy, but it is not a cure because of the latent reservoirs.
The resting memory T cells.
Right. Some of those infected CD4 cells don't actively pump out virus, they just go to sleep. They transition into resting memory cells with the viral DNA integrated safely into their own genome.
And why can't the drugs kill those?
Because the drugs only target active viral replication. If the virus isn't building anything or cutting anything, the drugs have nothing to interrupt. The virus is invisible to the drugs and invisible to the immune system.
Sleeper agents. Well, how long do those sleepers last in the body? If someone is on HAART and totally undetectable for a decade, don't the cells eventually just die off naturally?
They do, but the half-life of a resting memory T cell is incredibly long. They're designed by evolution to remember pathogens for a lifetime. Mathematical models in the source text estimate that it would take roughly 70 years of continuous suppressive therapy to wait for every single infected memory cell to die off naturally.
70 years.
So effectively a lifetime.
So if a patient stops taking their pills even after 20 years of being undetectable?
The drugs wash out of their system, a few of those sleeper cells wake up, and the virus rebounds to millions of copies within weeks. You have to stay on the medication forever.
Which brings us to prevention. We mentioned condoms, needle exchanges, and blood screening, but what about a vaccine? It's 2026 and we still don't have an HIV vaccine. Why is it failing?
It is the ultimate holy grail of virology, but it remains completely elusive. We've tried injecting recombinant envelope proteins like synthetic GP120 to train antibodies. We've tried complex viral vectors. Almost all of them have failed in phase 3 trials.
Why is this one virus so uniquely hard to vaccinate against?
There are 3 massive hurdles. 1 is the mutation rate we discussed. Quad-species is a constantly moving target. 2, the virus hides its DNA inside our own genome within days of infection. Antibodies floating in the blood can't reach a provirus hiding inside a cell nucleus.
And the third reason?
The third reason is arguably the biggest conceptual problem. We frankly don't know what natural protection looks like.
What do you mean?
In almost every other disease, we have vaccines for measles, polio, COVID. We have a natural model. If a human gets measles and recovers their immune system, shows us exactly what antibodies and T cells are needed to confer lifelong immunity, we just mimic that with the vaccine. Right. No human being has ever naturally cleared an HIV infection. We don't have a blueprint for what a successful immune response even looks like, because the virus has never lost a fight on its own.
That is a very sobering thought. Nature usually leaves us a breadcrumb trail, but here it just left us nothing.
Nothing but the CCR5 mutation, which isn't an immune response, it's just a missing door. However, we do have other prevention tools that are working.
Right, the microbicides.
Yes, topical vaginal gels containing antiretroviral drugs like tenofovir. In some studies, they showed a 39% reduction in acquisition.
And circumcision. The text lists this specifically.
Large trials in Africa proved that adult male circumcision reduces female-to-male transmission of HIV by about 60%.
60%, just from a surgical intervention.
Yes, because the inner foreskin is packed with exactly the kind of target immune cells, Langerhans cells and macrophages, that the virus loves to infect first. Removing that tissue removes the virus's preferred landing zone.
We have covered so much ground today, from the architecture of that bar-shaped nucleoid to the absolute tragedy of the unchecked epidemic in sub-Saharan Africa.
It really is a story of profound biological elegance and devastating human tragedy.
So to wrap this deep dive up, let's summarize the mission. HIV is a lentivirus, a slow, deliberate killer. It originated in primates and crossed over into the human population early in the 20th century.
And it uses a terrifyingly sophisticated array of accessory genes, things like NEF blinding the immune system and VIF destroying our cellular defenses.
It reverse transcribes its code, makes a million typos to evade our drugs, and hides its DNA inside our own chromosomes. It methodically destroys our CD4 command center, leaving the body wide open to opportunistic horrors like TB and pneumocystis pneumonia.
But humanity fought back. With HAART, with combination therapy, we've managed to turn an absolute death sentence into a highly manageable chronic condition for millions of people.
That is the summary. The science is incredible, But the fight isn't over.
Not by a long shot.
And here is the final thought I want to leave you, the listener, with today. The source mentions that even in patients who have been successfully treated for years, 1 million infected memory cells remain hidden in their body. Sleeper agents.
The latent reservoir.
Right. We have Timothy Ray Brown, the Berlin patient, as absolute proof that a cure is biologically possible if you are willing and able to completely replace a person's entire immune system with mutant stem cells.
Which we can't do for 35 million people.
Exactly. So the question moving forward, the billion-dollar virology question, is this: can we ever bridge that gap? Can we find a pharmacological way to force those million sleeper cells to wake up and show themselves so the drugs can flush them out without killing the patient in the process?
The shock and kill strategy.
Right. Or is that integration so perfect that this biological machine will simply always be a permanent part of the human condition now, something we can only ever manage but never truly defeat?
That is the defining medical challenge of our era. Science is inching closer, but as we've seen today, the virus is a brilliant moving target.
It certainly is. Thank you for taking this deep dive with us today. We really hope you walk away with a totally new appreciation for the staggering mechanics of this microscopic killer.
Keep asking questions. Stay curious.
We'll see you all next time.